I'm Tom Zinnan. I work here at the Biotechnology Center. I also work for the Division of Extension. And on behalf of those folks, thanks for coming to Wednesday night at the lab. Our other co-sponsors are Wisconsin Public Television, who's not here tonight, Wisconsin Alumni Association, and the UW-Madison Science Alliance. So we do this every Wednesday night, 50 times a year. Tonight, I get to welcome Audrey Gasch here to the Biotech Center. Audrey was born in
Chilton, Wisconsin.
Yeah. And you graduated from high school in?
1990.
No. What town? Sorry, that was ambiguous. And then where'd you go for your undergrad?
UW-Madison.
Oh, I've heard of it. And what did you study?
Biochemistry. But don't tell my colleagues in the genetics department.
And then where'd you go for your PhD?
I did my PhD at Stanford University.
And you postdoced?
In Berkeley, California.
Very good. And then you came here in 2004. Way to go. So she's now a professor in genetics. And of course, what's weird about the genetics laboratory here is that there's a department of genetics and a department of medical genetics. So one is in the College of Ag and Life Sciences, and the other is in the School of Medicine and Public Health. And it's about almost 110 years old now. The Department of Genetics, 1909. It's known almost about it. So, Audrey gets to talk with us about the new Center for Genomic Science Innovation, which she is the acting or founding director or interim director. I'm not sure exactly what you're. Good. I gave you a little handout. If you didn't get the handout, I'll see if I can go find some more copies. DNA is a pretty cool thing. It's a great alphabet. We got 6 billion letters in our genome. It's a lot of spelling. The patterns that we can see, the relationships that we can probe, that's all pretty cool stuff. It's great to study genes one by one, but it's also pretty cool to study genomes and to compare genomes across genera and families and phylums. And it's going to be interesting to hear Audrey's ideas of what this new center might do as far as innovations to speed the current tracks that genomic science is on, and maybe some innovations that might steer it in different directions. So would you please join me in welcoming Audrey Gasch to Wednesday night at the lab.
Well, thank you very much for coming out. It is really a pleasure to speak in this series. I know how much Tom puts into this series and the other outreach that he does, and it is really, really fantastic that this series exists, and I'm happy to be here. So I'm going to give, so I have heard this as a savvy crowd. I hope I have aimed it at the correct level, but I wanted to be inclusive and make sure that I make clear what genomics is. I'm going to talk about the history of genomics at UW-Madison and also the future of genomics, a little bit about our new center and where we're going, and at the end I'm going to talk in a little more detail about my lab's research. I'm happy to have an interactive discussion here, so if anybody has questions, feel free to raise your hand or shout out. I love that kind of interaction. So, as I think Tom said in his email, I'm a Wisconsin farm kid. I grew up on a hog farm in Chilton, surrounded by a sea of dairy farmers. And it is really true that I really, like a lot of farm kids, I think, was always captivated by biology. And I love this slide here that captures a lot of different organisms, showing them side by side. For many biologists, we are really intrigued by what makes organisms different from one another, but what are the common features that are really common to life. And so it's this great diversity in form and function that really, I think, took me from the farm through the path that you heard Tom outline. And so one thing that has been clear to biologists and humans for a long time is that the baby giraffe looks an awful lot like the parent giraffe. And that's different from the chicken. Yet there are still features that are in common between these organisms. And so when I was thinking about summarizing the history of genomics at UW-Madison, I'll introduce what that is in a minute, I realized that really the place to start was the history of genetics at UW-Madison. Genetics is the field of study, well, I like to think of it as the field of studying heredity. What is it that makes a baby giraffe look like the parent? And how does information get transmitted across generations? How does all that work? Well, that was the fact that there was heritable information that could be passed along generations was really first articulated by Gregor Mendel, some of you may have heard of, who worked with peas that had different characteristics, green versus yellow peas, wrinkled versus smooth peas. And he started to figure out that it was not random what the progeny of different crosses would look like. This is called nowadays selective breeding, actually. People can put 2 and 2 animals or plants together, and it is predictable to some extent what the offspring will look like. So in 1910, the Department of Experimental Animal Breeding was founded at UW-Madison and really was meant to capitalize on this knowledge to not just selectively breed plants and animals, but in the process really understand the fundamentals of how heredity works. And so this has been a very successful field. And in fact, this department was the forerunner to the UW-Madison Department of Genetics, where I'm a faculty member. And so in the old days, people realized that there was heritable information that could be passed on to the progeny. But it took a little while for scientists to figure out what the nature of that was. And as many of you have likely heard or studied, the nature of that heritable element is DNA. DNA is a chemical polymer. I think a lot of times people don't think of it as a chemical, but it's actually a series of repeating chemicals, a polymer. It is a polymer of 4 different chemicals that we call bases, and we have a shorthand calling those 4 bases G, A, T, or C. And it's the order and the sequence of many of these different bases repeated that has meaning to the cell. The genome, as you may know, refers to the total DNA content in an organism. All of the, to a first approximation, all cells in your body, in organismal bodies, has the same DNA genome. And we often describe this as a blueprint for the organism. I like to think of it as a DVD. And it's important to think about what use is a DVD. Is really actually not worth that much if you can't read the information. The DVD is not what's carrying out the function, it encodes the information. DNA is very similar. It encodes information that cells can read and information that cells can enact. So the genome contains all of the information needed to make an organism. And we now have the technology to read out the information, these 4 bases repeated over and over in different orders. We have the technology to read that genome of any creature now, humans, bacteria, mice, and I'm going to tell you a little bit about the history of that. Really, the big advance in reading long stretches of DNA came in the 70s. Of course, there were lots of technology and insights that led up to this. But a gentleman named Fred Sanger was one of the first to develop a new type of chemistry that allowed scientists to take a piece of DNA out of an organism and actually replicate it in a tube, so make a second copy of that DNA, and in the process be able to read out what the order of the bases was. And really, these advances enabled scientists for the first time to be able to actually read those 4 letters, G's, A's, T's, and C's, the order of those letters in different chunks of DNA. And Fred Sanger won the Nobel Prize. He actually won 2 Nobel Prizes. The one for sequencing came in the 1980s, which if you think about it was not, well, for especially by the end of my talk you will appreciate, that's not really that long ago. Because we've gone since that time from very laborious chemical test tube manipulations to read out a short chunk, maybe 1,000 base pairs at a time. From when I was a youngster in my undergraduate days, it was a lot of work to get 1,000 base pairs in a short order. Nowadays, we can sequence billions of base pairs in a matter of really hours. So here's what that looks like. This is actually sequenced from a gene from yeast, which is the organism that I work on. I'll tell you about more toward the end of my talk. And for those of you who are sitting close enough, you can see that this is a bunch of G's, A's, T's, and C's in different orders. So great. We now, starting in the 1970s, had the ability to read DNA sequence. The question is, what does this mean? And so in the early days, that slide was not that different from this in the 1920s. Hieroglyphics, what does it mean? Who knows? But having it is the potential to understand it. And I would say, just like with Egyptian hieroglyphics, having more of it, discovering more hieroglyphs, enabled historians to start to find patterns in those data and eventually make sense of what this language meant. And really, I think it was the same principles that influenced genomics, which is the study of genome sequences, and the study that is enabled by having a genome sequence, as I'll outline more in a bit. I don't expect you to see this, but or read any of this, but this is a timeline of genomics that was, timeline of a history of the Human Genome Project. This is really a preeminent journal that many biologists read. Many of you may have seen it, called Science. Some years ago, Science published a history of genomics, not all, but many of the important steps that led to the ability to sequence the human genome. All of the base pairs, 3 billion base pairs in the human genome. They started their timeline in the 50s with Crick and Watson, and also Rosalind Franklin, who helped study the structure of DNA. That weaves all the way through technological steps that enabled eventually the sequencing of the first genomes. The simple genomes were viral genomes, which were small enough that the technology could read all through the base pair sequences. And this really led to sequencing of more complex organisms that had bigger genomes. The culmination should be in quotes, with the first human genome sequence that was published in 2001. Of course, that human genome sequence that came out with a lot of fanfare was not complete. There were a lot of errors. But this was really a momentous occasion in the history of sequencing. We could be able to read the basis of a human genome sequence. I just want to point out, since I'm focusing on the history contributed by UW-Madison, that 3 of my colleagues are on this timeline from Science magazine because of their seminal contributions that really enabled the Human Genome Project. Dave Schwartz, who is one of my colleagues in our new center, CGSI, was one of the co-inventors of a new method at the time called pulse field gel electrophoresis that enabled scientists to isolate much bigger chunks of DNA than we had been able to isolate before. That enabled us to sequence longer pieces of DNA. My colleague Lloyd Smith is part of our center and a professor in the chemistry department, co-invented the first automated sequencers that could automate the sequencing of DNA so we could get much longer stretches of sequence at a time. And if you trace through this history, my colleague Fred Blattner, who's an emeritus faculty, and one of his students at the time, who's a current faculty in our center, Nicole Perna, sequenced the first bacterial genome sequence, the E. coli genome, which was a major advance and a predecessor of the human genome. I can't resist but highlight 2 other people who were on this list, because these were actually my graduate school advisors, David Botstein and Pat Brown. So in some sense, I'm the daughter of these genomicists and the sister of these guys, I would say. Faculty who are at UW-Madison made major contributions to this technology. So as I mentioned, the human genome is 3 billion bases long. So imagine this slide of G's, A's, T's, and C's that just goes on and on and on and on and on. The real question, though, is what does it mean? And this is where scientists are still struggling, I think, making advances, but there still is an enormous amount of the world of genomics that is not known. So how are we going to understand what these hieroglyphics mean? And I should point out, I guess I didn't say this explicitly at the beginning of my seminar. I should have had a separate slide. Why would we even want to know this? A lot of times biologists get lost in the mystery of understanding how cells work. And there are a lot of important insights that can come from just a basic fundamental understanding of how a cell or an organism works, how heredity works, why a giraffe looks different from a chicken. But, of course, what we also want to do is use that information to benefit society. And so there are many direct applications of understanding what these genome sequences mean. For example, one very important one that many of us are interested in is personalized medicine. If I could sequence your genome and understand what it meant, we could use that information to predict what your susceptibilities may be, to diagnose disorders, maybe at very early stages or before you even have symptoms, to customize treatments, drug regimes. We are just beginning to get in the realm of personalized medicine. I shouldn't say we're just beginning. We have made some advances. For example, some of you may know about warfarin treatment. Warfarin is a treatment that is a blood thinner. Many people who have had a stroke will take warfarin to prevent clots from happening. But it turns out a substantial fraction of the population has a mutation in their genome, a base pair difference in their genome. That makes them much more susceptible to that drug. So if somebody with that particular genome gets the wrong dose of warfarin, they could really keel over. If the next person who doesn't have that gene, has a different genome sequence, gets the wrong dose of warfarin, it will have absolutely no effect. So this is an example of something that we already can understand. We know how to look for that genome difference. We know how to use that information to be able to to decide what starting dose of warfarin a patient should be on. Should be on, yes. Yes?
Question about the definition of bases. The human genome is 3 billion bases long. What's the definition of a basis? Is that four?
No, sorry. Each base is, what I mean here is the position, and that position could either be a G, a T, a C, G, A, T, or C. Okay. So there are 4 possible chemicals that can be in this polymer. And what I mean by 3 billion bases, there are 3 billion positions. And so each of us has, a lot of our genomes are in common, actually. Very similar sequence from one person to the next. But there will be enough differences, and those differences influence how you and I are different in our behavior, in our medical susceptibilities, in our height, all kinds of things, right? Heritable things. Does that address your question? Yeah. So I just was talking about personalized medicine, but there are other applications of genomics too. And we are now in the realm, since we're in Wisconsin and I'm a farm kid, I don't know if there are any farmers here, but many dairy farmers actually are using genomics of their dairy cows to actually make predictions about which animals should be bred together and other types of analyses. So actually I would say in agriculture, in certain areas of agriculture, there's quite a bit of genomics that is understood and used in application. There are many other applications of genomics addressing the effects of environmental change or climate change. There are people who are using genomics to look at populations of creatures living in different environments and looking over time when environments change in different areas. Are there some individuals with particular genotypes that are more likely to survive variable environments versus getting wiped out in different environments? So all of and many other applications of genomics. So I should have made this clear in the beginning. This is why we want to understand how to read this language of 4 alphabetical letters, set of 26, so that we can not just understand biology, but have direct benefits to our world. Okay. So what does all of this mean? Well, DNA is a language on its own. I just mentioned the 4 letters of the alphabet. And so what that means is that DNA is not that different from other languages that we already may be familiar with. So I don't know how many of you can read this. This slide might not look that different to you from where you're sitting to the previous slide. However, languages have structure. And if I reveal the structure of that exact same text, you might actually be able to recognize this language because it is English. Languages have structures such as punctuation, periods, question marks. We know words are organized into groups, for example, sentences. We can detect very easily where a sentence is based on the capital letter of the first word and the period at the end of the sentence. There's organizations such as paragraphs and spaces. And so it turns out that the human brain really relies on this structure to be able to read the sequence. And so it turns out DNA sequence also has structure. It's just taking us some time to figure out what that structure is. So I'm not sure these colors show up very well. But what I've highlighted here, oh, and something is not animating correctly. Why is that happening? Well, I'll just describe it to you. I've highlighted this block here in magenta, and it might not be showing up. And you'll see that the very first 3 letters are dark blue and underlined, and the last 3 base pairs here are dark blue and underlined. This is actually what we call a gene. It's a real gene sequence, and it has a structure. Before I introduce the structure, let me just make sure everybody knows what a gene is. I think Tom had some very nice prose in the email that he sent out if you saw it. But a gene is really, I think of it as analogous to a sentence. It is a stretch of DNA sequence that conveys information. It has a start, it has an end. So, but the cell is reading this DNA a little differently than we read English or any other language. So here I have a cartoon of the DNA. Cells have machinery, which are made of proteins, which can scan that DNA and create another related molecule called RNA. DNA and RNA are really made of very similar chemical polymers, G's, A's, T's, and C's, little different for RNA. And so because these, but the difference is DNA is actually 2 of these strands that are intertwined. Many of you have heard of a double helix. RNA is actually just a single strand of this chemical polymer. But because these 2 chemical languages are quite similar, we refer to this process as transcription, like transcribing one written language into another. The cell then, for the most part, the DNA doesn't really do functions in the cell. And for the most part, RNAs are not doing mechanical functions in the cell. I'm oversimplifying. But it is actually the information encoded in the DNA and the RNA that is again read out by the cell, this time by a different protein. And now the cell will translate to a different type of chemical polymer called a protein. And this polymer is made of amino acid chemicals that are strung together. And so so the information in the DNA is transcribed to RNA, translated to a new language of proteins. And in many cases, it's the proteins that are actually doing functions in the cell. You'll notice I said the DNA is read by one protein, the RNA is read by another protein, and they're making yet other proteins that do functions in the cell. In genomics, so there are many, many scientists who study individual genes, individual proteins, individual aspects of this process. But in genomics, one of the things we would like to do is be able to identify all of the genes in a genome. That's one step in the genomic process. Does anybody have questions about this slide? People get the gist of what we're talking about here. So on my next slide, oh sorry, I'm coming back to this slide here. Here we go. Here's the start of our gene. Here's the end of our gene. Everything in pink here is part of the gene. How do we know how to recognize that? Because we scientists have learned enough about what the start looks like, what the stop looks like, and there's one other secret to identifying genes, and that is that the cell, this process of reading out that I told you on the previous slide, the cell reads groups of 3 base pairs when it's doing transcription, especially translation. So what that means is that when we have a new DNA sequence and we want to identify where are all of the genes, we computationally know what to look for. The start, the stop, and it better be a repeat of 3, because that's, without going into all of the details, that's how the cell reads it out. This is an opportunity for me to highlight another advance from UW-Madison, the biochemistry department. Gobin Khorana was one of the scientists who figured out this rule of 3 and how cells read groups of 3 bases to make proteins, and he co-won the Nobel Prize in Physiology in 1968. So great. We genomicists have figured out how to read all of those DNA base pairs and predict where the sentences are, the genes. Well, that all sounds great. That is a great step in our desire to understand how to read and interpret a genome. But here's a major problem with the human genome. It turns out only 2% of those base pair positions encode genes. In other words, 98% of those base pairs are not part of genes. We call it non-coding. So what are they part of? What do those base pairs mean if they're outside the genes? Do they mean anything? Are they functional? And where are they? Are there functional bases? What are they doing? And so we have a much harder time identifying, well, so I should say, it turns out, I'm going to introduce in the next slide, there are other types of important DNA stretches that are not genes. They're much harder to recognize computationally. We can't predict exactly what they are. And we're still trying to figure out what the functions mean. So what am I talking about? Well, here's another cartoon of a gene. I mentioned to you earlier, the gene is the sentence. It's a region of the DNA that encodes information. And the cell's going to scan that and read it, and in the process transcribe that to an RNA copy. Okay? So here's my little squiggle representing this RNA language from the gene sentence here. Well, it turns out not all genes are always making RNA and making protein. There are a lot of genes in our genomes that are reserved for specialized situations. Let me think of a good example. Well, my lab, as you'll hear about hopefully in a little bit, studies stress responses. So there are some genes that are dedicated to protecting your cells if you have a really severe fever. But if you don't have a fever and you don't need that function, it's wasteful for the cell to be putting a lot of energy into making RNA and making protein. So what that means is that a lot of genes are just what we call silent most of the time, and they get turned on under specific situations. And so there are little hidden signals in the DNA. I've annotated them as little colored boxes on this cartoon that are like switches, literally like a little, not literally, figuratively like a light switch, okay? And those light switches work, again, because we have proteins which are doing functions. When the cell senses the temperature gets too high, those proteins will scan the DNA, find their little light switch. Sometimes the light switch is in the gene, sometimes the light switch can be on the RNA. But what these proteins then do is either help turn the neighboring gene on so that it's making a lot of RNA and protein to do some function like protect your cells against a heat shock. Or in other cases, it can be shutting genes off so that they become silent. So these little light switches turn out to be really, really important for making a living cell. And so that means that there's much more that is important in a genome sequence than just the genes. So coming back to this cartoon, we have lots of little hidden sequences here. And genomicists are trying to figure out how can we scan this DNA sequence and be able to predict where those important light switches are, because we want to understand how the light switches work. Okay. So it turns out, I'm going to skip a little bit ahead here quickly, it's even more complicated than just reading the DNA base pairs, because it turns out now that we have had the capability to look at DNA in new ways, there actually can be chemical modifications to that DNA polymer. I'm not going to have time to go into the details, but what it means is that now there are entirely different proteins that can scan along the DNA and actually mark where the protein has been by adding a chemical to the DNA. That changes how the cell reads out the information. And it's even more complicated because it turns out, especially in recent years, people have figured out new technologies that allow us new views into biology. And now what we're realizing is that DNA is not just a sentence in a book. It's actually a three-dimensional chemical molecule. And that string, that polymer of bases, turns out, can fold up in very complicated ways. And that's important because now we're realizing some of these little light switches are really far away from the genes that they are controlling, which means, from a genomicist perspective, it's much harder to find which light switch goes with which gene. Okay? And because genes encode functional information, we would really like to know, for all of the reasons I said, personalized medicine, agriculture, etc., how do these switches work and how can we understand how they work and how genetic differences across individuals influence that. So how will we figure that out? So now I wanted to talk a little bit about the modern era of genomic science, just to summarize. So I've kind of gotten you up to the point here where we are today. Genome sequencing is very common and relatively facile. Most biologists use genome sequencing. If not generating a genome sequence, they'll download somebody else has already generated the sequence for their organism. So I'm not to say that there's not still room for improvement and technology innovations, but it means that the field of genomics has really evolved. We're no longer spending tons of money to just get the DNA sequence. We have tons of DNA sequences. We want to understand what it means. So the sequencing technology also has enabled us to identify, so we can now predict what are all the genes in a new organism. If we sequence a new genome, we can predict what the genes are. We can use the sequencing technology to actually also identify all of the RNAs that are in a cell at a given time. Lots of other technologies here. And so many of these technologies are generating massive amounts of data. So we have 3 billion base pair positions in the human genome. And so these other types of analyses that leverage having a genome generate massive data sets, these are just some pretty pictures to remind me to say that computational developments are really important in genomics. It's not just about the ability to generate the data, but making sense of all of the data is very challenging. And so computational development goes hand in hand and side by side with the technology development. Our real goal going forward, as I mentioned, is to understand how all of this data and genome sequence turns into a living, breathing, responding creature. I have a human listed here, but this is really a fundamental question that influences all aspects of life. And as I outlined with my precision medicine example, we want to be able to know if we have each person's genome sequence, how does that influence what makes us different? So it is in this light, this new era of genomics, that UW-Madison recently founded a new center called the Center for Genomic Science Innovation, or CGSI. And our mission is Well, it's right here. To develop new innovative technologies that allow us to see cells in new ways. Side by side, new computational genomic approaches that allow us to extract insight from that data. And really an integration of the new technologies and new computation to allow us to address new questions in biology that we were not able to address before. And we really want to aim that at modern problems in medicine, agriculture, industry, and really just basic scientific discovery. So I'm going to spend a few slides just giving kind of a broad overview of the center. And if there's time at the end, I'll talk a little bit more detail about my lab's research just to make it a little more concrete. So the center, as I mentioned, we were really kind of focusing on, well, I can describe it in terms of 3 different areas, but really a fundamental aspect of the center and really genomic research is that these things really are integrated. And so many of the faculty who are participating in CGSI span multiple areas, but for the sake of presentation, I've kind of categorized them into different groups. So one focus of the center is new technology development. And so I should say that the center really was just created. We formally came into existence September 1, about a month ago. We currently have 12 faculty members who are in the center and have space in the neighboring building. But the idea of this center is that we would really capitalize on some very unique expertise on campus and really, as one of my colleagues, Dave Schwartz, likes to say, provide a tent pole under which genomics research can really be enabled. And so our hope is to be able to bridge researchers in the center with many other researchers outside of the center at UW-Madison and beyond to really aim these new advances at important problems. So in terms of technology development, for those that I put primarily in this group, David Schwartz and Lloyd Smith were my 2 colleagues listed on the science timeline here. And so David is working on new technologies, new methods to be able to sequence DNAs and also synthesize very large DNA molecules. That better enables biologists to be able to test what happens if we change some of these bases, what happens in the cell if we do that. It helps us better understand the sequences. Josh Kuhn and Lloyd Smith are working on innovations in a technology called mass spectrometry. It is enabling the identification of different proteins and chemical modifications of proteins in cells, but on a global scale. So we can identify all of the proteins in a cell at a given time. Mike Sussman is one of the co-founders of a company people may have heard of called Nimblegen. That was acquired by Roche some years ago. And Mike has done lots of different technology applications. One of them is using new technologies to be able to synthesize DNA molecules for study. And also other ways of studying proteins and how proteins interact in cells. So the important thing about technology development is when we have these new technologies, it allows us to see biology in new ways. And this is really a critical advance to being able to understand cells. There's so much that we can't see in terms of how cells work. And so these new technologies are really important. Computational approaches really should be developed, and our focus in the center is to do this side by side, simultaneously. As new technologies are being developed, they create new types of data, massive amounts of data sets. So we need new computer algorithms that can handle that. We need new statistical tests that can extract biology. I won't list everybody up here, but UW-Madison has very strong strengths, very particular strengths in computational biology. And so many of those folks are in the new CGSI. And then, of course, one of the great fun things is to be able to take these new approaches, new computational methods, as they're being developed, apply them to biological questions. And it is actually it's one of the most thrilling things in my scientific life is getting to address questions that we haven't been able to address before. And so The biological applications come from many people outside the center, but in terms of people who are part of the center, it includes myself, Nicole Perna, one of the early sequencers of the E. coli genome I mentioned, and Mike Sussman, who really focuses on plant genomics and genetics. So I benefited in my early training from being one of in the right place at the right time. Those two other stars I highlighted on that timeline were my graduate advisors, who were amazing innovators in the field of genomics. And it was just thrilling to be a student in that era. And in CGSI, we aim to deliver the same things to our students. We have a very strong focus on education, in particular graduate education, highlighted here because we have two large grants from the National Institutes of Health that are dedicated to funding graduate students and postdoctoral fellows in becoming the genomicists of today and tomorrow. So one of these training programs is called the Genomic Sciences Training Program, directed by my colleague Dave Schwartz, again, one of those people on the timeline, and Louise Pape, who is the coordinator of that program. Another training grant from the NIH National Library of Medicine is CIBM, Computation in Biology and Medicine, directed by Mark Craven. And so these are really important, both for training, but also for really creating a very strong community at UW-Madison. I'm not going to go into this slide, but just to say that we aim to have connections with many different groups on campus. And for the sake of time, I'm maybe not going to go into this. If there are questions, not technical, but detailed questions about this afterwards, I'm happy to talk about it. But we don't want to be an island out there. We really want to connect many groups of people on and off campus. So let's see, I think I have a little bit of time if people have the stamina. I'm going to talk a little bit about my own lab's work. Also to make things a little bit more concrete, give you a little bit more of a taste of the details of genomics. So I had this cartoon before showing our DNA sentence, chunk of DNA that encodes information. That gene can be read by the cell and transcribed into this RNA language here that encodes information to make proteins. And I told you that many genes are silent until they're needed. And so if you get a really severe temperature, suddenly a bunch of genes get activated. And now if we could look into the cell, we suddenly see the RNAs that came from those genes are present. And if we could use Josh Kuhn and Lloyd Smith's methods, mass spec methods, we could identify all of the proteins from those genes are suddenly in the cell. So now this is very useful for genomicists because we want to understand what that genome DNA sequence means. But now if we can say, well, we have a normal body temperature, we can identify which genes are on or off. And now in heat shock, whether you have a severe fever, lo and behold, different genes turn on. Now we can start making hypotheses. Maybe those genes that turned on have something to do with protecting your body against heat. That gives us a bunch of new ideas to then go follow up on. But simply identifying genes and proteins doesn't really make a cell. We'd like to be able to identify all of the proteins that are on in the cell at a given time. We can do that now with genomic approaches. But lists of objects also don't make a living, breathing cell. It is the dynamic interactions of all of the entities in the cell. The DNA, how those light switches are being activated, which RNAs are being made, which proteins are being made. Of course, there are many other levels of physiology I'm leaving out. But these are all chemical molecules. They're not floating around in a bag, scientists are realizing. Cells are very, very organized to enact functions. And this is what, these dynamic interactions are what give rise to living, breathing, behaving, reproducing creatures. And so if we're really going to figure out how to read a DNA sequence, we also need to understand how all those parts make a cell. And really, living creatures are more than just the sum of those parts. So we need to think about dynamics and interactions. It gets very complicated quickly. And it's really complicated to do this in humans, for those of us who are interested in personalized medicine. So many of us use what we call model organisms. My lab studies environmental stress responses in yeast, budding yeast, Saccharomyces cerevisiae. That's a big fancy word you may or may not have heard of, but you probably all experienced Saccharomyces cerevisiae because it's the same yeast that people use to make bread. It's the same type of species of yeast you use to make wine and beer. It turns out that the biochemistry and the genetics of the lowly single-celled yeast have a lot of fundamental features in common with how human cells work. Now, of course, yeasts are much simpler. We don't have to worry about different tissue types. We don't have to worry about cell-cell communication. Those are super interesting questions. Yeast would not be a good model if you wanted to study those things. But what we're really interested in studying is what are the fundamental ways that the yeast genome is read out by the cell? How do all those light switches work? How does that protect the cell if the cell gets shifted to really stressful conditions? And so yeast is an excellent model to do that with. The yeast genome is much, much smaller than the human genome. So that already is a simplification. It's a single-celled creature, which is a simplification. But yeast cells, although we think of them as being domesticated because they're used in industry to make bread and wine, turns out that this species of yeast lives in many different environments out in nature. And that means that these cells are very good at surviving sudden environmental stresses, sudden environmental shifts that can be stressful. Sudden heat shock, exposure to toxins, being caught in a downpour suddenly changes all the nutrient availability and the osmotic conditions. Yeast cells are very good at surviving starvation because they probably experience it a lot in nature. And so what that means is that yeast are a great model for studying how cells respond to environmental stresses, but it also turns out that studying environmental stresses in yeast is itself a great model for bigger picture questions in how genome sequences are read out and how genomics works. So some of the questions that we ask in our lab are these. How do yeast cells monitor their environment? How do they know when there's a problem in particular? Now, yeast are very simple, but we have enough tools to be able to look inside the cell or assess what they're doing inside the cell to know that when cells get a really severe dose of stress, like a heat shock, for example, they will change many different aspects of physiology. How do they do that? How do they coordinate that? Many different changes. How do they transmit a cellular signal that actually coordinates that? An important thing that we've realized is that when cells get really stressed out, they generally stop growing for a little while until they fix the problem or acclimate. And again, to make the point that model organisms are models because other creatures do this also, it's maybe not a surprise. In a drought year, the corn is all this high, right? When plants are stressed, when humans are stressed, when bacteria are stressed out, they usually stop growing to try to redirect their energy to fix the problem or address the stress. How does that happen? How does that regulate it? We are also really interested in looking across different individuals that have different genomes. And so how do different isolates of yeast, they're all the same species, but yeast from Malaysia have different genomes that are we can tell they're from Malaysia, they're the Malaysian type, from yeast that are from Wisconsin oak tree soil, which we've collected from our farm. And how do these responses evolve? So my lab has really specialized in an area called transcriptomics. So I've used the word genomics a lot today, which means the study of the study of and studies that are enabled by having a whole genome sequence. You remember I said that transcription is the act of going from the DNA chemical to the RNA chemical. And I already mentioned that identifying when a gene is on and making RNA and when it's silenced can be informative if we look across many different conditions. So the field of transcriptomics is a field enabled by having a genome sequence where we can look at every single mRNA in the cell and see how does that pool of mRNAs change if we give cells a severe heat shock, or we starve them, or we give them nicotine, which we have done just to study these responses. So what I'm showing you here, without going into the technology, turns out we use similar sequencing technology. We can also sequence the RNA polymers. I won't go into that, but what I have here is a cartoon of sets of RNA molecules that came from 3 different genes. I hope you can see the colors. The green gene is on to a little bit in the cell. This is before stressed, an unstressed yeast cell. We grow them in liquid culture. They're just floating around, plenty of nutrients, nice temperature. They're super happy. And so there's a green gene. It's kind of on a little bit. The cell is scanning it, making some RNA copies of that gene. Here I have 1, 2, 3, 4 RNAs that came from the green gene. Now we have, meanwhile, somewhere else in the genome, the blue gene. That one's really cranking, that light switch is really on. It's cranking out lots of copies of itself, okay? Same sequence, just different molecules. Then there's an orange gene. It's really not doing much. There's 1 RNA made, and it's kind of mostly quiet or silenced. But now let's say we give this cell a heat shock, just to stick with our example of heat. We take this flask of yeast and we stick them in the oven. We heat them pretty good. They're not dead, but they're stressing out. So now what do we see? The green gene, now we let them sit for a little while after heat shock, and we see enough time to respond. How do they respond? What do they do? These cells are probably freaking out. They're desperately trying to live and hang on. The green gene, about the same copy as before stress. Not much happened. Maybe no light switch effect by heat shock. By the orange gene, boom, the light switch got flipped on by the heat shock. Now we see tons of the orange gene transcripts, meaning lots of transcripts are made. We infer from this that the orange gene is turned on because the orange protein that's going to get made is really important for trying to address whatever problem the high heat is causing to cells. Now you might also have noticed the blue gene was cranking out lots of mRNAs before stress, and after stress, it got silenced. Why? These are kind of things that we make hypotheses about. So I'm showing, and so now we can quantify the relative change, the relative amount of each RNA after stress versus before stress, after the heat shock. So this orange gene there's 4 times more of it after the stress. So we infer from that the cell induced or flipped on the light switch so that there's 4 times more of the mRNA. The blue gene, actually, comparing after heat shock to before, there's 1/4 of the amount. So we infer it must be getting shut off. This guy, no change, not affected by heat shock. In the diagram I'm going to show you next, what we can do now is colorize these, because now I'm showing you just 3 genes. Yeast cells, they're so simple compared to humans. Humans have 20,000 genes. Yeast cells have about 6,000. That still is a lot, let me tell you. So, but it turns out that the human brain is very good at picking out patterns. And so one of the things, and actually I should say that my graduate advisor, Pat Brown, was one of the people who really pushed this technology of transcriptomics. And he is a brilliant person and realized that the human brain is very good at picking out patterns. It's really hard to read 6,000 numbers, but it turns out if you convert those numbers to colors and you organize the colors, patterns immediately jump out. And this is a diagram of those colors. Let me see if can I dim this? Let me see, dim stage. So this is a colorized diagram of yeast transcriptomic data. So each row represents a different gene, okay? So we have 6,000 rows. Each one is a different gene. And And for each individual gene, I've colorized it like I did on this previous slide. Maybe I didn't spell this out. If the transcripts went up in abundance after stress, you get a yellow color. If the gene gets shut off and the transcript goes away, you get a blue color, okay? And so we think of these as being really important during the stress. And maybe these are important to get rid of. And so this is what it looks like when you have 6,000 genes, but I have more than just one heat shock experiment. This diagram summarizes hundreds of individual samples from cells responding to different types of stresses. We looked at DNA damaging agents, temperature shocks, oxidative stress, osmotic stress, starvation for different conditions, and since then we and others have looked at thousands of other conditions. And so these colorized, and the data have been colorized now. So we don't have to look at a table of millions of numbers. We're looking at colors, and you might think, well, how does colors actually help you? Well, and I just have a couple more slides actually that I'll go through. I'll just finish this part up. You might actually notice there are blocks of colors. So hopefully you can see that there are patterns in this data. And the reason there are patterns is that the order of the genes is not random. We use a computational method, a computer algorithm called hierarchical clustering. Turns out to be pretty simple computational method and very, very powerful. And the crux of this method is that it doesn't change the data at all. It just changes the order of the genes such that genes that have the same behavior in response to many different stresses are all grouped together. So what that means here is that you see a group of genes. This here is about 300 genes. Each row is a different gene. But the colors across here indicate that these genes are grouped together because many of them get turned on, that's what the yellow means, in response to lots of different stressful conditions. Does that make sense to people, even if you don't fully understand the diagram? Then you have this block of genes down here. Remember, blue means that the gene was getting shut off. This is about 600 rows. Each row has pretty similar pattern, and that's why they're grouped together. When you see the whole group of genes, the pattern really jumps out at you. There is a group of them. These genes are getting shut off in response to lots of different stress conditions. So, what does that mean? In my last slide, I just want to bullet point some things we've learned from doing this. What can even just from looking at the patterns, we've learned a lot. One thing that we know is that when yeast cells experience different types of stress, DNA damaging toxins, heat shock, starvation, those are all pretty different. They pose different challenges to the cell. And so what we find is that if we look closely at all of the genes and how the cell's responding to a given stress, there's always something specialized about the response. Without going too much into the details for the sake of time, there may be some conditions. This little block of yellow here represents a group of genes, not much color in response to these columns that represent the response to different types of stress. But here's a block of yellow. These genes, their light switches are turned on specifically in the stresses that correspond to nitrogen starvation and amino acid starvation. What does that mean? Well, it turns out these genes are really important for synthesizing the cell's own amino acids. The cell can actually synthesize its own amino acids, but if they're out in the media to soak up, why waste the energy? You can just suck them up from the media until we shift the cells to broth that has no amino acids. They suddenly realize they're starving, and they invest the energy to turn on all these genes to make their own amino acids. That's what that block of yellow is. So we've learned a lot about. Now we can go back to the genome and say, oh, we know the sequences, and we know which genes these are. Some genes we might not have known what their functions are. But we see that they're all being turned on with the amino acid biosynthesis genes, other genes where we know the function. We scientists can now make a hypothesis that that uncharacterized gene that has that behavior probably has something to do with amino acid biosynthesis. Even though there's always something specialized about the response, I highlighted this big block of yellow and this big block of blue. These are genes, these genes here, that have a common response. There's certainly subtle differences, but these guys are getting turned on in response to many conditions. These guys are getting turned off in response to many conditions. So a large part of the response is actually a common stress response that we called the environmental stress response, and my lab is still studying what the purpose of this response is. Okay. Last point I will make is, lo and behold, so we first got these data in 2000, right, in 2001, right around the time when the human genome sequence, the first sequences were coming off the pike. We had this data from yeast. And now, since that time, other people have looked at stress responses in other organisms, and lo and behold, other organisms have similar responses. They have commonly induce a bunch of genes that turn out to be involved in protection and stress defense. They repress other genes that turn out to be involved in growth and growth control. And so this is an example of why yeast is so powerful as a model organism, because it's much easier for us to study this, to get insights, but yet we can relate those insights to other organisms. It doesn't mean it absolutely happens that way in other organisms, but in many cases it does. And so that gives the human biologists a starting point. So in closing, I hope that this seminar was enlightening for you. We touched on a little bit about UW-Madison history. We touched a little bit about what is genomics and the history of genomics, a little bit about my research, and a little bit about this center. It's an exciting time for us. We have a whole new realm or time in the field of genomics. And so we look forward to making new advances and seeing how the field evolves once we make those advances. So with that, I will thank you for your attention. I'm very happy to take questions or stick around for private discussion afterwards. But thank you. Are there any questions? Yes.
Yeah, kind of a trivial question. So you have DNA is double-stranded.
Yes.
And a messenger RNA is created. Does it matter which strand gets duplicated, or is there differences? If one strand gets demonstrated, it's a different messenger RNA and it has a different function?
That's an excellent question. And so actually, let me bring up my slide. The question is, since DNA is a double helix and RNA is a single strand, does it matter how the DNA is being read? And it does. I don't have a good diagram, but I'll work off of this cartoon. So I mentioned that we have proteins that can come and scan that DNA. Turns out there's not just one protein, there are a bunch of proteins involved in this process. And one of the processes is to unwind that DNA. And actually, one of those strands is the template to make the RNA. And it absolutely matters which template is used. And so those light switches that I talked about actually will orient these proteins so that they know which strand to use as the template to make this RNA, and essentially which direction to read out in. Does that address your question?
Are they related? Is the messenger RNA here related in some sense or other to if the other strand is read instead? Do they have a relationship? Obviously, there is a relationship between the two strands to begin with.
Yes, that is a very excellent question. So the question is really how are the two strands of the DNA helix read out? And if you make RNAs from both of those strands, are they complementary? That might be a paraphrasing. Yeah. So I skimmed over this a little bit, but let me back up and just say the DNA, actually, I mentioned that there are 4 bases in this chemical polymer. It turns out that the double helix is actually like a puzzle. So that the 4 bases that we read, the G's, A's, T's, and C's can be read out on one strand. And it turns out there are what we call complementary bases on the other strand, so that the helix can come together and wind up as you see these pretty pictures of DNA. So when the RNA is transcribed, those get unwound. The RNA molecule that's made is based on the order of the DNA polymer molecules. And so if the cell were to make an RNA from both of those templates, the two RNA that were unwound, the 2 RNAs would themselves be complementary. So it would be possible for those 2 RNAs. RNAs can actually bind together, not quite in the same way that DNA can. So this might be getting overly complicated. There are some cases, normally when the cell is reading a DNA, it knows which strand of DNA, of the unwound DNA, to use as the template. There are cases actually where the other strand can be read out in a way that regulates how much of the first transcript is being made or how much of the first transcript is being translated. That's an active area of study, I would say. So I described how mRNAs can be used to make protein. In some of these cases, the cell will make RNAs whose job is not to make a protein, it's to try to block the function of this mRNA because they can complement, they can bind together. Does that make sense? Enough sense, yeah. Any other questions? Yes.
So if you've been making a protein and then under some stress response we stop making it, do those proteins die out? How does their overall number decrease?
Yep, that's an excellent question. So the question is, if the cell has made a bunch of protein, as I have on my little cartoon diagram here, and then suddenly we shift to a stressful condition and that gene gets shut off, so there's no more new protein being made. It depends on what happens to that existing protein, depends on what type of protein it is. In some cases, if the cell is trying to get rid of that protein, let's say it's detrimental to surviving a heat shock, the cell wants to not make any more and get rid of what's existing, the existing protein. In those cases, the cell may have mechanisms to go in and gobble up the existing protein and get rid of it very quickly. In other cases, the cell may actually decide that it doesn't want to make any more, I'm anthropomorphizing, but the cell may actually stop the production of new protein, but still want to hang on to the existing protein that's there. So in other cases, those proteins, even though the gene gets shut off, the production of new protein gets shut off, the cell will actually stabilize the old protein so that it will still persist and function, even though there's not new replacement protein coming. Does that make sense? Yeah. Yes, Tom.
So it was 20 years ago next month that the Nibelgen paper came out. There are, on occasions, you have a glimpse of what you think is coming next. 7 years ago was CRISPR. Do you have a hunch of 2 or 3 or 4 things that you're hearing about that might be the next pretty interesting thing that's going to shift the direction of where we're going?
The next, well, I can tell you a couple of things that are already changing the face of things and still under development. One is the miniaturization of these technologies, some of the technologies that I've kind of touched upon, such that we can, so I described how my lab is using yeast and studying transcripts in individual cells. But really, we have not had the technology to look into individual cells. We've had to scrape a whole bunch of cells together and to get enough material to sequence. And so that means we're kind of always looking at the average of the population. So one big thing that's kind of already on the scene, but just dramatically changing, is technologies that allow us to do single cell measurements. So now we really can take an individual cell. We can identify, it's still, the signal to noise level is still challenging, but we can do it. You can identify all of the transcripts in an individual cell. That technology is already here, but other single-cell approaches are also really being advanced and still under development. Single-cell methods to identify the three-dimensional structure of DNA, that's going to be another, I think, really big, already under development, but emerging. And then there certainly are new ways of sequencing. Even though I downplayed sequencing, there are new technologies that are really changing the speed with which we can sequence, which is really important if you start thinking about human, personalized medicine, where you need a human genome, in the NICU, they want it immediately, right, for diagnosis. We also want to be able to sequence organisms out in the field for climate change research or agricultural research. So there are certainly technologies in sequencing that are completely different technologies that are really changing the face of things. And then I really think, I may be biased because I'm focused here, but computational approaches to interpret the genome are really, I think we're going to have major breakthroughs in that area and continuing development, but it still is a major issue. We have lots and lots of data from all different types of technologies. The technologies are changing, we have more and more data, but we still need to figure out how to put it together to understand what makes a cell. And so I think methods to be able to integrate those data sets are going to continue to be super important. Is there another question back there? Yes.
About the latency of the DNA, and so you have the shock. Does the DNA change by the time you make the measurement?
I'm sorry. Under the heat, the stress conditions.
Yeah. So you stressed it.
Yes.
And then there's a latency after the stress for you to make the measurement.
Yes.
So what happens in the DNA in that time by the time you measure it?
Yeah, that's a very good question. So the question is, when we do these stress experiments, we expose the cells to some stress, but of course there's some processing that takes some time. And I think your question is, well, how is biology changing in the time that we can even just collect and make the measurement? Is that the
correct?
Yes, that's an excellent point. And so that is one. Let's see, I just lost my train of thought here. You raise a really important point. And in fact, let me just say as a side note, the ability to do this on a transcriptomic level, to look at all RNAs in the cell, actually made scientists realize that the methods they were using to process their cells was actually completely stressing the cells out. They were responding big time to that. And nobody really realized cells are living creatures. They're no dummy. If you take the cells, remove them from their nutrients, spin them down in a tube, wash them with a salt buffer. Put them on ice. This was the common way of collecting yeast cells. They hate that. They have massive responses to that. So in fact, it was the genomics, the initial genomic studies made us realize, we should not be doing this. But your point is well taken. So when we do our experiments, we try to actually kill the cell as quickly as possible. So we shift them to the high temperature. And then we have a way of killing them that keeps everything intact to the best of our knowledge. But this is, when I talk about technology development, there are many things we can do now, many aspects of physiology we can glimpse at. But there are always room for technological advances, and some of them actually have been in instruments that allow us to kill cells quickly and freeze them. But the other approach is actually to watch living cells dynamically. And so these are other approaches. Tom mentioned other things that are going to come on the field. Microscopy methods, new ways of looking at minuscule structures and watching cells over time is maybe a better way to be able to follow some of these things. And those will be technologies that continue to emerge. Yes,
so what are these
Tom?
little guillotines that you're using to quickly kill them?
Well, this is another really good question. So Tom asked, what's the guillotine we use to kill the cells? we always had used freezing, what we call flash freezing. You can take cells in a tube and drop them in liquid nitrogen. You stick your finger in liquid nitrogen, your cells will die, and that's what frostbite is. It turns out in yeast cells, in the laboratory strains of yeast, the genomes that are reflective of the lab yeast that we use, if you drop them in liquid nitrogen, they completely die, and they're frozen in time, as far as we know. So that's great. We used that for many years. We can give cells a stress and then quickly drop them in liquid nitrogen, freeze them. But then we started, I mentioned this fairly quickly, we started thinking about, many people who use model organisms will use individual stocks or strains, a particular genotype, genome sequence, that is very amenable to laboratory conditions. For decades, people have been using the same strain of yeast. Of course, and we can freeze, we can save them, we can store them, we can get them out of the fridge and use them, they're still living. We can send them to other labs. And so over time, everybody doing yeast studies, we're really using mostly the same strain of yeast. So in our lab, we and others at the same time started realizing, wow, yeast cells live in nature. How different are the ones in nature? We're studying environmental responses. We were making all kind of, we were making all kinds of inferences about what cells do in the wild when they're exposed to heat shock. And at some point it occurred to us, we actually should check a wild strain, right? So we started doing this and realized the power of yeast for personalized medicine. We were doing personalized yeast stress responses by looking at yeast from Malaysia, yeast from Wisconsin, yeast from the Bahamas, yeast from Siberia. Each one of these populations got a little bit of a different genotype, little different genome sequence. And so it turns out that the guillotine we had been using in the lab, which is freezing them to kill them, we discovered doesn't always work. Most strains, if you drop them in liquid nitrogen, they'll die, just like the lab strain. But lo and behold, Tom, which strain of yeast would you guess is the one that survives the freezing and thawing?
The Bahama one.
It turns out you're actually correct, but no, it is the Wisconsin yeast can survive freezing and thawing. And would you guess, one of the Bahamas strains can survive freezing and thawing. But when we sequenced its genome, so Bahamas, we collected the yeast strains, and I should say this was a former undergraduate in my lab, Dan Quiddick, who collected these yeast from his family farm. The Bahamas strain that we got was from a strain collection. Other people will collect strains and put them in. We can, if we freeze them in the right way.
That's not very reliable. I think you personally need to go into that.
That's true. Well, anyway, we were surprised that one of the Bahamian strains could survive freezing and thawing. Well, wow, this is what happened here. We sequenced the genome. From the genome, we could tell it was a Wisconsin genotype. We don't know that, yeah. So we made lots of jokes about Dan tracking soil from his farm to the Bahamas. But that is the power of genomics. We could read the genome sequence. We can't understand all of it, but we could certainly say, whoa, the differences in this genome match the differences that this strain came from a colder climate. And in fact, it had the characteristics of that strain because it can survive freezing and thawing.
So then how'd you make the measurement?
How did we make the measurement if we can't freeze them? We have to be super careful. Actually, depending on what we do for that strain, we sometimes use very high doses of ethanol, which can kill them. It's not perfect. But yeah, so this is, sometimes we're limited when we do research and we make the best of what we can. And we try very carefully to always keep that limitation in the back of our minds when we're interpreting the data.
So what's a high dose of ethanol for Wisconsin?
The question is, what's a high dose of ethanol for Wisconsin yeast? Yeah, 151 proof. Other questions?
It's a federal highway.
Any other questions? Yes.
It
seem that you've got a lot to learn yet. So, if we're looking at Star Trek Next Generation,
Yes.
Dr. Crusher replaces Lieutenant Worf's spinal column. That's 23rd century. So, will we know by then how all of this works?
By what? Sorry, what was the date?
23rd century.
23rd century. You know. So the question essentially is, when are we going to have this figured out? I really don't know. I think that's a very good question. One of the things that is true to science is we set out with a set of questions. And as we make progress answering those questions, we suddenly realize all these new questions emerge. So my feeling is that we may never have it figured out. Because just when we answer the questions that we're setting out to answer now, whole new realms of biology and the physical world and how biological systems interact, I think will become apparent. Things we never anticipated. So I don't know when we're going to figure it all out.
On the other hand, there are people who think they haven't figured it all out.
Yeah, that's true. Now, it might be easier to say, when will we be able to take a patient genome and customize their treatment based on that genome sequence? And that is going to be, and already actually we're certainly in more experimental domains, we are in that phase.
That's essentially what Dr. Crusher did. Worf's genome made a spinal column.
Oh, I see. You mean taking a genome sequence and making synthetic tissues based on that genome. I don't know. Spinal columns is not where we're at. But, you know, we, yeah, I wish I knew more about the field, because I probably, the fact is we are making tissues out of patient samples already. For sure that is true. Because now we know enough about how to take an adult's cell from skin and actually revert that cell back to a form where it can be now redirected to develop into different tissues. That we can already do. Making a spinal column is probably a little bit of a high hurdle for where we're at right now, but that is actually, you know, there's all kinds of stem cell treatments that people are using now. So that actually is going to be, we're already there, actually, taking patient sample and genome sequence and making tissues and treatments from that.
So 23rd century might be okay.
Yeah, maybe so. We might be okay.
Any other questions?
Whoa, Roger.